All right, in Unit 4.4, we are concerned about how membranes are able to help our bodies maintain homeostasis, okay? And one of the ways that they do this is by transporting things across the membrane. So the question here is, how are substances transported across membranes? So first of all, small molecules like water can diffuse across a membrane. Remember that the membrane is made up of a bunch of phospholipids that are crammed together like people at a concert, but there are spaces between them. So if a molecule like water is small enough, it can fit through those spaces. Okay, there are other larger molecules like salts and proteins. Here are some proteins that are embedded in the membrane. They are a lot larger than water and they cannot fit through. They would be like a beach ball bouncing off the membrane surface or somebody's body surface. Okay, if they're too large, they cannot move through the membrane. Okay, so the type of transport shown by these molecules that are moving from a high concentration on this side to a lower concentration, this is called diffusion. Okay, diffusion, I don't know if I can draw in here. Nope, I cannot. So diffusion, D-I-F-F-U-S-I-O-N, is the type of transport shown by these molecules. Okay. So let's talk about some of these molecules that may or may not make it through the membrane. Okay. When we make lemonade, it seems like a random transition, but we'll get there. We need to have a solute, and this is the lemonade powder. Okay. A solute is anything that can be dissolved in a solvent. The universal solvent is water. There are other solvents like paint thinners and sometimes alcohol, but water is known as a universal solvent because it dissolves a lot of different things. Okay, when you put solute and you dissolve it in a solvent, you end up with a solution, in this case, lemonade. Okay, so those are some new words. Make sure that you understand those. In this diagram, we can see that we have 3 different solute concentrations. Okay, keep thinking about the lemonade. Our solute concentration would be very low here, maybe even 0%, and a medium solute concentration and a high solute concentration. This glass of lemonade is going to have more solute than these two. Okay, this is going to have very little solute. Now, we need to be able to look at the cell and say, now, is this solution, this very clear lemonade, does this have a higher or lower solute concentration than the cell? And we can say that these look to be equal, and this one has a higher solute concentration than inside the cell. Okay? This matters because water will move across the membrane because it is a small molecule, but its movements are dictated, they're controlled by where the salt is higher. Okay, so osmosis is a new term. This is specifically the diffusion of water. Okay, it's a type of diffusion and it's focused specifically on water. So osmosis means water moving in and out of the membrane, and how it moves, how it diffuses, depends on the tonicity or the saltiness of the solution around the cell. Okay, now we can have 3 different types of solution. Okay, first of all, we can have a hypertonic solution. Hyper means more than, right? Hyperactive kids have more or too much activity. Okay, hypertonic means more salt in the solution. Okay, we are always going to describe the solution with these terms. We never describe the cell. Okay, hypotonic solution. Hypo is the opposite of hyper. This means not enough or lower. I don't love this example, but one term that people are familiar with is like a hypodermic needle. That means underneath the skin. Okay, so if you ever have to get anyway, there are certain things in medical science where they put a needle underneath your skin, and that's using a hypodermal needle. okay, hypodermic needle. So hypotonic is less salty than the cell, okay, the solution that is less salty, and isotonic is going to be equal saltiness, okay, inside and out of the cell, they are equal concentrations, okay. Now, it's important to remember or to note that solutes, those salts, are large molecules that cannot pass the membrane, okay. So if there's a lot outside the cell, or if there's a lot inside the cell but not outside the cell, these are not going to pass the membranes, okay. So in a hypertonic solution, okay, so you have, wish I could draw on this right now. I don't think that's going to work. In a hypertonic solution, you have more solute, more salt outside the cell. The water is going to move out of the cell. Remember, a lot of the cell is water. It's going to move out towards the saltiness to equal the solute concentration, or the tonicity. In a hypotonic solution, it's going to do the opposite. If a hypotonic solution, you have, say, 1% inside the cell and 0.1% outside the cell, the water is still going to move towards the salty, but in this case, that's going to mean it's going to move into the cell, okay? Again, trying to equalize the concentration of solutes. Isotonic, you still have water passing the membrane because it is able to, but you're not going to have a lot of water moving in or a lot of water moving out. Okay, it's already balanced. It is at homeostasis, exactly where we want it. Okay. So we did this in class a few times. It wouldn't hurt to practice this. What kind of solution is the cell in each of these? Okay, give yourself a few seconds to think about it. Go ahead and pause the video if you want. Okay, this cell has an equal concentration of solutes inside the cell and out, so this is an isotonic solution. Okay, the middle one here, the cell is in a solution that has less salt out in the solution than inside the cell, so this is going to be a hypotonic solution. This cell is in a solution that has more solute than inside the cell, so this is going to be a hypertonic solution. Again, we only describe the solutions. We never describe the cell by hypertonic, hypotonic, or isotonic. Okay, so let's take a look at this. The cell is 1% salt in all cases, and that's roughly what it is in our bodies. And it's sitting in a 10% salt solution. This is a hypertonic, right, solution. Right here, this is going to be lower than the cell, so this is going to be hypotonic solution. In this case, the cell is sitting in a solution that has equal amounts of solute. This is going to be a isotonic solution. Okay. Now, let's go back to this. In this case, we need to think through then, which way is the water going to move? Is it going to go into the cell or out of the cell? Water is always going to move towards the salty. So in this case, water is going to leave the inside of the cell and go to the outside solution towards the salty. It's going to try to equal out this imbalance. Okay. Here, the water is going to move into the cell because even at 1%, this is still saltier than the outside. Okay, so the cell will actually grow. Okay, it'll fill up with water and it's going to get bigger. In this case, right, we had water leaving the cell, so this cell is going to shrink. And we're talking about animal cells here. In this case, nothing's going to happen to the cell size because we're in an isotonic solution and it is balanced, so no big change in water content needs to happen. Okay, now we talked about IV fluids, right? If you're dehydrated and you go to a hospital and they say, oh wow, you've had the stomach flu, or it's summer and you haven't been drinking enough water, we want to rehydrate you. They are going to hook you up with an IV bag of fluid, okay, intravenous fluids, and this is not straight up water. This is not 0% salt, okay. This is going to be actually 0.9% saline solution, because that is what our cells are bathed in, okay. Our body water is actually kind of a solution, right? It has salts, it has glucose, it has different things dissolved in it. Now, if we were to get IV fluids that were just water, we would end up with our cells sitting in a hypotonic solution. Okay, so kind of like this middle one here, right? That would be very bad. Okay, what would happen to the cell here? We would have water entering the cell, and eventually it would burst. Okay, so that's the same scenario that's happening here. Human blood cells, they contain roughly 0.9% salt. If you were to place these blood cells in pure water, 0% salt solution, the water would enter in through osmosis and the cells would burst. Okay, I would encourage you to draw this out. Use a little cup, give yourself a solution, give yourself a cell, assign percentages. Make sure that you understand the diagram, and then ask yourself the question again. Okay. So in your notes you have these different things. We are not going to cover facilitated diffusion, and we're not going to cover bulk transport. But we are going to talk about active transport, and this, I want you to put in your notes, is a form of transport that requires energy in the form of ATP, and it also requires a membrane protein. Okay, here we see a protein in the middle of our phospholipid bilayer. This protein is going to be able to work as a pump, but it needs ATP. Okay, so you need a protein and ATP. So active transport, here's an example of this. This is the movement of solutes against the concentration gradient. So here we have just 3 solutes on this side and a lot over here, but the proteins are going to continue to pump solutes onto this side, into the cell, because the cell needs a higher concentration of them. Okay, you see how if this were diffusion, these guys would be spreading out onto the other side, but this is why active transport is usually against the concentration gradient. Okay, what is needed? We need ATP energy, and we need a membrane protein, one of these guys, to act as a pump. The ATP will give the energy to the protein to do the pumping, right? If you're pumping something, you require energy to do this. Okay, so here's an example of a question that you might see with active transport. This cell requires more of a particular solute inside. Inside the cell we have 8%. Outside the cell we have 4%. Okay, so we need some of this to go inside the cell. We are working against the concentration gradient. It is unnatural, right, for the 4 on the outside to move in, okay, because there's already more in here. They should be trying to equal out. They're not. We need the cell to have a higher concentration of solute. So what form of transport is needed for this to happen? This would be active transport. Okay. What is required for this process? We need ATP energy and a membrane protein. Okay, so how are substances transported across the membranes? Well, there are 2 different ways. Okay, we have passive transport and diffusion, in which small molecules can move freely through gaps in the membranes. This does not require energy. Okay, this is passive. An example of this is osmosis, which is the diffusion of water. Okay, the other way that things are transported across the membranes is through active transport. Okay, this is a process in which larger molecules can be moved with the help of proteins. It also requires ATP energy. Okay. Go.